In a quiet Cambridge laboratory, a drop of cells began to do something extraordinary. Within 2 weeks, those cells turned red. Not from dye, but from blood. Human blood, made entirely in the lab. At the University of Cambridge, scientists have created three-dimensional, embryo-like structures that mimic the earliest stages of human life. They call them hematoids, from the Greek word for blood. These tiny, self-organizing structures behave like miniature embryos, and after just 13 days, they begin producing blood stem cells, the same kind that give rise to every blood cell in the human body. The scientists started with human stem cells, cells that can become anything. Under the right conditions, those cells built themselves into complex layers: ectoderm, mesoderm, and endoderm, the three building blocks of all human organs. By day 8, something remarkable happened. Heart cells began to beat, and by day 13, red patches of blood appeared, visible to the naked eye. Each hematoid can produce blood stem cells that transform into every blood type: red blood cells, white blood cells, even specialized immune cells like T cells. This breakthrough could help scientists understand how blood forms naturally inside embryos, a process impossible to observe directly, since it happens deep within the womb at just 4 weeks of life. It could also help us model deadly blood diseases, like leukemia, and test new drugs safely outside the human body. The cells used to create hematoids can be made from any human cell, even your own skin. That means one day, doctors might grow perfectly compatible blood for each patient. No donors required. Unlike previous techniques that need complex chemical cocktails, this new model grows naturally, letting the cell's own environment trigger blood and even heart formation. Dr. Jitesh Nupane, who led the work, said, "It was an incredible moment when the dish turned red. We could see the blood forming with our own eyes." His colleague, Dr. Geraldine Jowett, added that hematoids capture a stage of blood development that could open new doors for immune research, even cancer modeling. And Professor Azim Sarani, the senior author, believes this is only the beginning, a step toward regenerative therapies that could use a patient's own cells to repair damaged tissues. These embryo-like models can't develop into real humans. They lack key tissues like the yolk sac and placenta. Still, every experiment follows strict ethical review before proceeding. The research has now been patented through Cambridge Enterprise, protecting this powerful tool for science and medicine. From a few reprogrammed cells, to a beating heart, to human blood flowing for the first time in a dish. What began as a spark of curiosity could one day transform medicine, and perhaps teach us more about what it means to be human. If you love exploring the future of life and science, subscribe to LG News, where discovery never stops evolving.